How we doing today? Yeah. Live and kicking. Questions? Surely there's questions. You're like, I'm sure. Really nothing? So we have a test coming up when? Next week? Next 2 weeks? October something? 2nd? What day of the week is that? All right, 2 weeks from today, basically. So one question was, what would it look like? And my sarcastic response was a piece of paper with questions on it. But that's what it will look like. I mean, it'll be questions about gene expression, questions about embryonic stages, questions about the processes that we've learned so far. You won't have. Yes, 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 yes. Any question type you can imagine could be on there. Not made it yet, I don't know. And what difference does it make? None. Makes zero difference. Makes zero difference. Do you believe it does? I know you do, deep in your heart, believe that if you know there's a true-false question, that now you know something about the test. But it's a false sense of security. It doesn't tell you. Today we want to keep talking about differential gene expression, and probably we'll finish this off today. So we mentioned last time that enhancers and promoters work together, and that promoters kind of generally let the gene be turned on, but enhancers and silencers can cause tissue-specific regulation. So they could limit the genes being expressed in a certain tissue or activated only in a certain tissue. And that's, of course, again, going to be the tissue at question is going to need to have that particular transcription factor in it, and the place on the DNA where it can bind, and whatever combination of factors that might exist are going to have to be there. So this is a couple of examples of gene regulation for a couple of different genes. So this one is the crystalline gene, which is expressed uniquely just in the lens of the eye. And this is the somatostatin gene, which is expressed primarily in some cells in the pancreas. And so the gene itself is in here, and there are promoter elements, and the promoter elements are similar for both genes. They have a TATA box, which is this, where TVP or TF2D binds. They have SP1 boxes. I don't know, I guess this one doesn't, but this one does. But it also has an enhancer area down here, and this has an enhancer area upstream. So remember, enhancers could be almost anywhere in the DNA. They will have to be on the same chromosome, and I mean they have to be reasonably close, but they can be pretty far away, like 20,000 base pairs, which is, you would think, well, that's really far away. But many genes are quite large, so it's actually less distance than a gene. And this just shows some of the different proteins that can be expressed there. All the ones that are on the top have a positive effect on transcription, the way this book has drawn it. And any they draw on the bottom has a negative effect. So these two proteins bind kind of to the same spot. And if this one is here, it cancels out the binding of this one and turns that gene off, actually. So that one negative can actually rule out all of those positives. And these are all positive, right? Some of them are the same. There's several of these PDXs, and then there's several PBXs, and a CREB, which is a CEVP binding cat box. This is a cat box. We mentioned that. So combinatorial regulation is something to always think about. Multiple transcription factors working in combination. Not just one transcription factor. Vast majority of genes need lots of transcription factors working together. Working together might be working all for positive stimulation of expression, or it might be also looking at negative regulation. Okay, so competition. Here is some data to show you exactly how we can analyze this. And I think I mentioned before how you could attach a reporter gene to a promoter or an enhancer. So the normal gene, you want to have the normal gene there. If you jack with the normal gene, you might disrupt the formation of that tissue. And then how can you study the genes expressed in, say, the lens of the eye if you have caused the lens of the eye not to develop, right? So you have to always think about is the effect that your experimentation is having, is the result that you see a result of what you changed directly or indirectly, right? If it's an indirect result, then you can't draw as good conclusions from it. So with a reporter gene, you have left the normal gene intact, and you've simply added the expression of another gene, which is not normally in the cell, but shouldn't cause any problem with the cell because it doesn't do anything that should bother any cell. For example, beta-galactosidase or the LAC-Z gene from bacteria. There's really no consequence for this gene to be produced or the protein to be expressed in any cell. It just makes an enzyme. It's a harmless enzyme. However, if you dump some substrate in the cells for that enzyme, it can go ahead and cut it and give you this blue color, okay, which shows up. Now, at this point, when you're observing this embryo has already been fixed, okay, it's dead. It was removed. This is a mouse or a chick. Probably a mouse, since those are mouses. It's been removed from the uterus, and its development has been stopped by putting it in formaldehyde, and the cells have been fixed. So all biological processes are stopped, and you're just looking for the protein, and the protein's enzymatic activity can still work. And so you're looking at where blue color develops, which is it's supposed to be blue down there as well, which shows up a little better on the real picture, but not so good here. That's the limb bud. And this is, I guess, where the eye is going to develop. So that particular gene is supposed to be being expressed in the lens. So they've been using the lens enhancer to drive the expression of the beta-galactosidase gene. And it is showing that, yeah, it's mostly expressed there. And they can do this in other places. So here, for example, is the expression of a reporter gene, again, the LAC-Z gene, being driven by the normal promoter for this gene called L1. And this gene has an enhancer, but it's actually a silencer in this case. This silencer prevents this gene from being expressed in cells that aren't part of the developing nervous system. So here's the normal expression pattern with the intact regulatory sequences, and here it is with that sequence removed. So the promoter in this case lets it be expressed everywhere without any sort of regulation whatsoever. It's just, hey, you're on, go for it, you can be transcribed. And so it's expressed in the limb bud, there's a limb bud, there's limb bud, it's expressed all throughout the notochord, it's expressed in the somites, it's expressed in the neural tube where it's supposed to be, neural tissue, but it's expressed in a lot of other places. These are parts of the head here called the branchial arches where it's not supposed to be. And here's where it's normally supposed to be, and that structure, which I'm going to guess is probably the ear, and then down the back here in the neural tube and in the somites. That's where it's supposed to be. And those are probably neural crest cells that are becoming the peripheral nervous system. It's not actually a cell line. But this can give you the example of how a silencer element can regulate to limit where a gene is expressed. The promoter is kind of like a permissive thing. Expression can occur, and enhancers augment that expression in certain tissues, and silencers can restrict that expression to certain tissues. Okay? So you can see how transcription can be studied, and how we learn about this is largely by putting these genes as reporter genes along with the transcriptional control elements back into an embryo. You're creating in that instance what's called a transgenic organism. And we can probably talk about the two sort of levels of transgenesis. So one would be putting the gene in as an exogenous gene and that means you're putting that gene in extra. It's not, wouldn't have ordinarily been there, and you're putting it in without changing the endogenous gene at all, okay? So you retain the endogenous gene. So these are terms that you'll hear. Exogenous, which means from the outside, and endogenous, which means the one that was already there. So the endogenous gene is retained, and you're simply adding, okay? You're adding a trans gene, getting it expressed. Now that's going to get incorporated into the genome. So when we say transgenic, we mean that this DNA that's being added to that cell is being incorporated into that organism's chromosomes. But we're not targeting it in any respect. It could just go anywhere in the chromosome. And that can be problematic. It might go into a place in the chromosome that turns out to be heterochromatin. So even though we put something in there, it's not expressed because it's in a region of the chromosome that's not expressed. So you have to worry about these kinds of experimental data. And you need to, when you get a pattern like this, you have to go in and show where your sequence is and has it actually been altered, or is it in a weird place in the chromosome? Have you lost anything else about it? Okay, so that would be a non-targeted, we call it non-homologous. You're not putting it in the same exact spot, right? Then there's homologous. You're going to target that. You're going to target your transgene. You're going to put it in the place where the gene that you're interested in is, and at the same time, you're going to be eliminating the endogenous gene. Okay, so this is a replacement. You're going to replace the endogenous gene with the exogenous gene, okay? So if you were to do it with this, then the endogenous L1 gene would be replaced by this. Now remember, you have two, we're talking with diploids, so they got two copies. So you can generally get away with doing this because they still have the wild type copy that provides normal function, and now you're just putting in a second copy in the same place. Putting it in the same place means any sort of chromosome structure, compaction, heterochromatin structure that might be there it can be ruled out. You can control for that now because it's in exactly the same environment that the regular gene would be. These are called knockout. Okay, so if you replace the endogenous gene with an exogenous construct that's not that gene, like if you switched out the normal gene for the gene LAC-Z instead, you've knocked out the gene that was there and replaced it with this gene, okay? And of course, if you create a homozygote, Now if that animal completely lacks that gene, you've created a total knockout. And now you can look at what that gene might have been doing in the animal based on phenotypes that might exist. Like maybe the limbs don't develop properly, or the nervous system doesn't develop properly without this gene involved, for example. So transgenic, and we'll get into more detail of how it's actually done, but transgenesis is a type of experimentation strategy that you just need to know in the back of your mind right now, so it's not mysterious, okay? It can be done. You can replace an endogenous gene with an exogenous piece of DNA, and you can target it to go to exactly a certain spot in the genome. And they've gotten really good at it. So if a tissue or a cell type is controlled by hierarchical sort of transcriptional regulatory events where one gene may be activated in an early cell type along a developmental pathway. So if we think about the pancreas in this example, as the pancreas is developing, there are going to be pancreatic stem cell sort of cells. And those cells are going to be pluripotent. They're going to have multiple potentials. They're going to have the ability to form multiple cell types, but not every cell type. They're going to be restricted to the kinds of cells that would be found in a pancreas. But as they differentiate, they're going to now narrow into more restricted channels. So for example, if the gene PDX is activated, that's a transcription factor, and we find that it's activated in pancreatic precursor cells. So if a cell is expressing that transcription factor, it will pursue one of these cell types. If it's expressing the transcription factor NGN3, which is a second transcription factor, and everything that's expressed above is continued to be expressed throughout. Okay, so this is on first, but then this is on. These cell types that express both things are now restricted to only 3 of the potential cell types. They're restricted to the endocrine cell types that are in the pancreas. Whereas if they don't, they become exocrine cells, which are secreting like amylases and nucleases and proteases and stuff like that. Stuff that gets secreted into the digestive tract. These are going to get secreted into the bloodstream, right? These are endocrine. If it expresses a third transcription factor called NKX6.1, they're quite inventive with the names, aren't they? Then it will be restricted to a beta cell, which are the cells that produce insulin, for example. And after that is expressed, a mature beta cell will also express the factor MAF-A. So those 4 genes need to be expressed in order to turn on all of the things necessary for a cell to follow down the beta cell, the insulin secreting cell fate, right? So could you take these genes and turn them on in a non-pancreatic cell pardon me, and cause that cell to be re-specified down a beta cell route? The answer is yes. So for example, they've created a viral vector that expresses those 3 genes, this one, and this one. I guess you can bypass that one. That viral vector was then injected or infected into a beta cell deficient diabetes mouse model strain. Okay, so these mice were engineered to lack beta cells. Okay, and so they have to inject them with insulin, the little life of a little mouse, or restrict their diet. In any case, this is a common use thing as well. Mouse model systems. Okay, they didn't just find a mouse laying out on the street somewhere and test him and say, oh, yeah, this little mouse has diabetes, let's raise it. They made the mouse. They made the strain of mice, engineered it. So when they put this back in, lo and behold, after a few weeks, if you look at a biopsy of their pancreas and stain it for cells expressing insulin, you see that they are expressing the vector, they've got the viral protein in them, and they are expressing insulin. And so anytime it's yellow, they have both. But you also see that sometimes it's red without having yellow. So this has somehow, perhaps, the viral vector has been lost from some of the cells. But there's a few cases where cells are blue, but they're not making insulin either. So perhaps the viral vector was defective and it didn't express that gene. But in a pretty significant amount of time. The other thing is this: you can feed the mouse sugar and it doesn't die anymore because it's not diabetic anymore because it's cranking out insulin. You see the benefit of that? If you're diabetic, you do, right? Could you restore through a viral vector? All they did was infect the pancreas. It infected non-beta cells, maybe it infected these other endocrine type cells, and switched them to the beta cell phenotype. Okay. Now, what do you have to worry about? Well, with humans, you have to worry about what was that virus, and will the body have some kind of crazy reaction to the virus, which would then attack your pancreatic cells and just kill them right back again because they're viral infected cells. Your immune system might go, oh, you're a virally infected cell, and he's like, no, but I'm making insulin. It's like, screw you, boom, you're dead. No more viral infected cell, right? Because the body says that's an infected cell. So you have to worry about the immune system. So you have to pick the right vector. You have to pick something that our immune system sort of ignores. And you can do that by making engineered viruses. Usually they use adenovirus. But that's hard to do on certain tissue types because the virus is restricted for what tissue types it can be infected into, right? So you have to play those games. And you can't just transfect a pancreas. You can't just dump DNA in there and it gets taken up into the cells. It has to be carried into the cell through some sort of a vector. All right, so that kind of wraps up transcriptional regulation. But you can see there's a lot of cool stuff that goes on at the transcriptional level, right? So the next level of regulation is we call it post-transcriptional. So it's what happens to the RNA. So the biggie is differential splicing, okay? All messenger RNAs get capped, and they get polyadenylated, and there is some regulation that happens with the poly A tail, and there can be decapping and so forth. But probably the greatest level of differences come through what we would call alternative splicing or differential splicing. So you have one gene here outlined in the genome with the introns are colored, and the introns can be removed, but you can use or not use sets of exons. You don't have to use all the exons. So in this case, they skipped the yellow one and the blue one. In this case, they skipped the green one. So you're always going to put them together in order. So you're never going to go red, green, yellow, blue, purple. You're always going to go in order from first intron to last, from first exon to last exon. And the first and last exons will be always part of the RNA. So you won't have an exon that isn't, that you won't ever splice off the first exon. If you don't want the first exon, you have to not transcribe it. If you don't want the last exon, you have to also not transcribe it. So that means you can start in alternative places and you can end in alternative places. So to make different kinds of RNA, we have three choices. We can have an alternative start, alternative splicing, or alternative termination, which we call polyadenylation. So you can either start at a new place. So if you didn't want this red exon, you'd have to start transcription in here somewhere. And the first nucleotide that's transcribed becomes the first nucleotide, the first exon. If you didn't want the purple exon, you'd have to stop before the purple exon. Okay? This would require a different promoter because presumably there's a promoter here. So if you're going to start a new spot, you're going to need a new promoter. The promoter designates where you start. And if you're going to terminate, the reason this terminates here is because there's a polyadenylation signal. So if you're going to stop over here, you need a second polyadenylation signal. And you need a way to use the two different promoters or the two different poly A signals. If you're going to alternate in using of exons, that means you're going to have alternative sequences within the introns that let the splicing factors cut the exons out differently. So you're going to have to either have different promoters, that's the alternative start. You're going to need different splice sites for alternative splicing. And you're going to need alternative polyadenylation sites for alternative endings, alternative termination sites. Okay? All three things exist in a multitude. As a consequence, we see that you need fewer quote-unquote genes. You need fewer parts of the genome that are saying, here's a gene, because one gene could be used to make multiple RNAs from starting in different places, splicing in different places, and stopping in different places. And each RNA, once that RNA is made, it's just going to be translated. So the RNA in us, if the RNAs aren't altered again after they're spliced, they're used. So they make a different protein. But it basically means that that one gene in the genome could be responsible for 3 or more proteins. Okay, not just one, which means the number of protein forms we have in our cells is greater than the number of genes. So I said this, cell type. So if you want to have a gene that's expressed in one tissue and you want to use exon 3, and in another tissue you want to use that same gene, but you don't want to use exon 3, you're going to need something to tell the splicing machinery what cell type it's in and how it should use those exons differently. So you're going to need cell type specific or tissue specific splicing factors. These are not your general splicing factors. Okay? He wants to show off and say they remember what the splicing factors are from genetics. Golden opportunity. Not to know all of them, just in general. What does splicing? SNRPs. Yep. And what is a SNRP? It's an RNA, right? It's a small nuclear ribonucleoprotein. So it's an RNA that base pairs. Well, you can't just use those. You'd have to have a different one to recognize a slightly different sequence, or you need a protein to help it. And so a lot of the splicing factors are proteins that make the SNRPs bind at different places than they would ordinarily bind. And a lot of times what they're doing is they're sort of covering up one spot so the SNRP goes over here because it's like, I can't bind to the best spot, so I'll bind to the next best spot, right? So we would call that perfect spot, you know, a consensus or a canonical site, and we would call the other spot that it wouldn't normally use a cryptic site. It's sort of masked or it's hidden. It wouldn't ordinarily use it or see it, but if a protein is there, it could sort of point to it. Think of it as like one of those pictures that they have on the internet sometimes. Is like, can you see the rabbit in this picture? Can you see the snake in this picture? Or can you see the, I always think of the Monty Python skit. You guys ever seen that? It's hilarious. Well, it's not hilarious. It's hilarious, but you're not supposed to laugh at this stuff anymore. There's like a bush over there, and they're like, you know, Mr. Johnson has hidden somewhere in this thing. We don't really know where he is. And so they ask him to stand up, and he stands up, and then they blow him up. And then the next time they don't stand up, but there's only 3 bushes, so they just methodically pop the 3 bushes and they get them. So they're showing you how not to be seen. So that's cryptic, right? How not to be seen. So you have a sequence of bases that's not perfect for the SNRP, but if you point it out, you go, hey, look right there. You could almost bind to that. Why don't you bind there since you can't bind to your good spot? Okay, and that's how it works. You have these splicing factors that cover up the good spot and at the same time help them bind to the not so good spot, and all of a sudden you get a different exon. You get an exon not used, or you get an exon used that wouldn't ordinarily be used, right? A lot of people think is exons are going to get used automatically, but a lot of genes, the normal sequence would just pass right by that and not actually use it. So you have to make it be used, okay, to get all the exons used. So whenever you make an RNA, you have to make it encode, it'll encode a different protein. Some RNAs would encode a non-functional protein as a consequence. That's true in some of the Drosophila examples that we'll show here in a minute. Like, here are some examples. So you've got a precursor that's made for collagen, a precursor that's made in chondrocytes. Those are the ones that are going to make like hyaline cartilage. And you have another precursor that's made in chondrocytes, but these are made in the early chondrocytes when they're creating collagen, but they need to stay as chondrocytes, and then these are made in the mature ones. Here's two different alternative uses for, this is the 3' acceptor site, so it either goes there or it goes over here. And then here's for FGF receptor. So this receptor was expressed in the limb, and this is expressed in the mesoderm. And they bind to different FGF molecules, different growth factors. This is a fibroblast growth factor, FGF. Then the R means it's the receptor for it. So this hooks onto that. Here's for cordon. This is the normal cordon, and this is a non-functional version of cordon. Cordon is a gene that's going to be involved in notochord specification. Hence the name cordon. Okay, so lots of genes are alternatively spliced, and they maybe have 2 alternate forms, but some have many alternate forms. Troponin. Troponin is a gene in muscle cells. Of course, you have 3 different kinds of muscle cells. They all use troponin, but they actually use different isoforms of it. Different mRNAs are made. Plus, there's troponin made in neural cells, and there's troponin made in some other kinds of cells. They're all different. Here's 3 genes called sex-lethal, transformer, and double sex that are involved in sex determination in fruit flies. And there's a splicing pattern that is kind of, you would say, is default, and one which is caused by specific factors. In this case, there's a female-specific factor that allows this gene to be spliced so that 1, 2, and 4, 5, 6, and exon 3 is omitted, because when exon 3 is included, there's a stop codon in frame. So this makes a non-functional peptide. It makes a peptide, but it's a new thing. This makes an actual protein. The protein, in turn, helps make that same splicing pattern happen, and it causes the transformer gene to be spliced in a female-specific manner, where exon 2 is left out, and guess what? Exon 2 has a stop codon in it. Having a stop codon is not the norm. This just happens to be 2 examples of it, and it's kind of coincidental, but it is just a coincidence. And then transformer, guess what it is? It helps also alternatively splice this protein, this gene called double sex. And this makes a different protein in both males and females. And what happens when you have this protein present in males at the same time is they have both male and female hermaphrodites. They have both characteristics and vice versa. So these 2 proteins act to turn on specific characteristics of male or female development in the fruit fly. So if this one is expressed, if you put this gene in a male, it kills it. If you don't have this gene in a female, they die. So that's why it's called sex lethal. No matter which sex you are, if you're supposed to have it and don't, you die. If you're not supposed to have it and do, you die. Because it's interacting with other genes. It's not shown. But that's an example of how differential splicing can regulate a whole developmental program, in this case, sex differentiation in a fruit fly. It's not how our sex determination is made, but it is how for fruit flies. And that's just more about it. We need to go into that. That's still more about it. Okay, So another thing about, if you're really super cool interested in fruit fly sex for some, we won't talk about it, reason, you can look at those other slides, but I'm going to jump into it. Another thing about the messenger RNAs that can be regulated is how long do they hang around? How long do they get translated? So how much protein gets made? Because the RNA is there a long time, more protein, right? If our RNA gets translated and immediately degraded, then you might only get one protein molecule made from it. So stability of the RNA, which we often describe as its half-life, we don't really measure its half-life, but we use that same terminology from physics and radiation. But how long is the RNA hanging around? And the stability of the RNA can be mediated, or we have found, is often mediated by specific sequences. So any RNA molecule is inherently less stable than DNA molecule because it's single-stranded. The extra hydroxyl groups are reactive. The extra hydroxyl groups on the sugar, remember, are reactive, and there are a lot of enzymes. Like there's a couple of enzymes that degrade DNA, and they're mainly only made in the pancreas. They're for the purpose of degrading DNA that you've eaten in your food. And RNases are in there as well, but every cell has lots of different RNases, and those RNases can chomp down on RNA sequences and destroy them. So the cell goes to some measure of energy expended to cap the RNA, to put a poly-A tail, and both of those structures stabilize the RNA. Okay? But other ways can be to bind the RNA with proteins, which would cover up sequences that would be recognized by the RNases that would be destroying the RNA. So you can mask those sites and protect the RNA. A lot of RNAs, when they're translated, the ribosome knocks that protein off, right, as it comes through. And now the RNA is vulnerable and it's immediately degraded. So it basically gets to get translated one time. The histone proteins are this way. So when a histone protein gets translated, it's it. It gets translated once. And to make another histone protein, you need another histone RNA. Okay? Maybe it gets translated a couple of times. There'll be a couple of ribosomes there before it gets degraded. But it barely makes any protein before you have. So that means transcription and protein concentrations are tightly coupled. If the RNA can hang around for a long time, you could make the RNA once. You could transcribe the gene one time and forget about it because the RNA is going to be there for another 2 or 3 hours making a buttload of proteins. Okay? So if you want to tie transcription regulation to protein concentration, you've got to have a short half-life on your RNA. The RNA has to go away quickly and only be translated a few times, so that there's a correlation between RNA amount and protein amount and DNA making the RNA. So here shows a sequence that in this is the wild type sequence of the mRNA, and you can see that it's pretty AT rich. This is not the poly-A tail. This is in its 3' untranslated region. And this RNA is degraded pretty quickly, as you can see. And here's a control RNA that's not being degraded. And then they mutated this to change most of the T's to something else. And you can see that that RNA is now quite stable. So this is for beta globin, I guess. This RNA is destabilized by that sequence. That sequence actually recognizes a nuclease enzyme that starts chomping down the RNA as just after it's translated. All right, so a lot of messages are stabilized in this way, and that stability is regulated. So if you look here, they're using the term half-life, and they're looking at the presence of some proteins over here. Like these proteins, vitellogenin, are yolk proteins. When we think of yolk, like the yellow in a chicken, you're like, ah, it's yolk. But what is yolk? Well, it's actually a protein called vitellogenin and ovalbumin and things like that. And so here, these proteins are made by the liver. Okay? So in the liver is where the RNA is being expressed, not in the egg. And the protein is secreted into the bloodstream. Protein is absorbed by cells in the ovary, and the protein is then transported into the developing oocyte as protein. Okay? So if the frog is supposed to be making eggs, that would normally be under the regulation of a hormone like estrogen. And if you treat the cells with estrogen, you get a stable half-life. 500 hours is the half-life of the telogen RNA. What does that mean? It means the gene transcribes one RNA, and 500 hours later, half of that RNA is still there. Okay? It's a long time. If you don't have estrogen, 16 hours later, half that RNA is left. Right? So it degrades quite quickly in the presence of estrogen, in the absence of estrogen. Right? I don't know what that fold difference is. 30, 10, 15? I don't know. This is 3 times different. This goes from 22 to 2.5. This is in chicken eggs. Same thing, kind of thing. In mouse, the protein casein, which is made in mammary glands. With prolactin, very stable. Without prolactin, not stable. Okay? So the hormone is activating a series of events. How is the hormone stabilizing that? Well, it's activating stabilizing proteins that bind to the RNA. So they mask the RNA and protect the RNA. So stability of the RNA is an important thing to think about in development. Polyadenylation. So RNAs get polyadenylated, and I just said part of that's for stability, right? But the poly A tail on an mRNA can also be a great handle because it's hanging off out there and it's not sequence specific, so it can just be bound by things. So there are a lot of proteins that just bind to the poly A tail. Specifically, poly A binding protein is one. So there's not just one poly A binding protein. So we found out, we thought there was, but we found out there's actually tissue-specific poly A binding proteins. And there's developmental stage-specific poly A binding proteins. Okay, one of them, for example, is the protein SMOG in fruit flies. So the gene nanos, which is German for dwarf, causes the embryos, when that gene is mutated, the embryos are shortened and they lack posterior structures. Okay, so nanos, short embryo dwarf, nanos is regulated or controlled by SMOG. So the whole Hobbit thing, right? Dwarves, the SMOG, the dragon. Yeah, the fruit fly people are awesome. They make up names that you can remember a story by. What the hell that the protein does, but at least it makes a cool story. So anyway, SMOG binds that. A lot of mRNAs are polyadenylated when they're transcribed, transported out of the nucleus, and immediately deadenylated, not entirely, but largely removed. So I think the rule of thumb is polyadenylation in the nucleus, co-transcriptional polyadenylation, will put on somewhere around 750 to 1,000 A's during that time interval that it has. So messenger RNAs are transported out of the nucleus with approximately 750, let's say 1,000 A's. But then that's often trimmed back to just a few, like 30 or 40 A's. And under that condition, it's stable, but it's not translatable. So that prevents, that short of the poly A tail prevents the RNA from being translated. So here the RNA is perfectly stable, it can hang out, but it won't be translated. So you can create a messenger RNA stockpile without making a bunch of protein because you don't have room in the cell for all that protein. So a lot of oocytes, a lot of eggs of organisms like frogs do this, fish do this, chickens do it a little bit, but not much. We don't really do it at all. And a lot of invertebrates, like fruit flies, create tons of stored RNA in the egg. And that RNA is just there to be translated at a later time. Okay, so these RNAs are, they're adenylated and deadenylated. So when I say it's not polyadenylated, write this down. It is polyadenylated, but it's been deadenylated mostly. It's never totally not got poly A tail. The poly A tail is extremely short, and it blocks translation. And then, when you want it to be translated, it gets re-adenylated. And that would be by a cytoplasmic poly A polymerase. That would be tissue specific. So maybe just in the liver, or the liver precursors, or maybe just in the mesoderm, or maybe just in the endoderm, right? So by putting the poly A tail back on, boom, now it can get translated. So it's like a switch, a timer. Okay? So how can RNAs be regulated? Give me the broad strokes. Because you're asking, what would a test look like? It would look like broad strokes. Give me the broad strokes. How can you regulate an RNA? Alternative splicing. Alternative start sites. Alternative poly A site, termination sites. Masking of splicing sites is how you do the splicing regulation. Alternative promoters, how you're going to get alternative starts, right? Polyadenylation, that can regulate translation of it and stability, right? So you can regulate its stability, you can regulate timing of translation. So you can regulate the actual RNA itself that's produced, where you start, where you stop, what exons are in it. Requires alternative factors, right? So you would know what you're doing and how you got it. Alternative promoters, alternative splicing factors, alternative poly A recognition sites. Then you would say, okay, stability, that's mostly 3' untranslated regions that are responsible for that. Every now and then there's a wild hair and they have a 5' untranslated region that's involved, but 9 times out of 10, if you're going to the casino and pulling the handles, this would be your bet. If they say, what is the polyadenylation stability site? You go 3' UTR, and it's going to cash out every time. So, and then polyadenylation, okay? Does that make sense? This is an example of something where the poly signal is used differently, just to be crazy about it. And here shows you some RNA getting stored. I don't know which one this is. Nanos. So nanos is being translated, but then nanos is being bound by an anchor protein, which is SMOG, and a whole bunch of other things that are named for dragon stuff. And then here's one for where the protein the RNA gets translated, the protein gets degraded unless the protein is, or the RNA gets degraded unless it's interacting with its master protein. So this is a stability example. That one was just a trapping. This one is a stability. These guys get destroyed. These guys get to live. Nobody's getting translated yet. How do they get to the end? This makes it look like that there's they have brains. They don't. How do they get to the end? They're actually being carried there along microtubules. And so some get carried to one end and some get carried to the other end of the microtubule, which is going to be polarized inside any cell. You're always going to have a plus end and a minus end. So some go to the plus end, some go to the minus end. Like Oscar goes to the minus end, or goes to the posterior end. I don't remember which end of the microtubule it is. And Big Boy goes to the anterior end in fruit flies. Okay. Stability. How do you actually get an RNA to be degraded is sometimes in a cool way through microRNAs. Okay, microRNAs. They can base pair with the sequence and cause that sequence to be targeted by a nuclease complex called RISC. So acronym, we'll stop here. RNA induced, induced, I think, stability complex. So that binds to these double-stranded regions. You have a untranslated region of this RNA. It's the Lin 14 RNA, and the Lin 4 RNA is a small RNA that has several places that are homologous to it in this gene. So the Lin 4 RNA is the, I think, the blue one, and that is the different spots. And so it can base pair in a lot of different ways, but it can form base pairs, at least with pretty good amount of them. And every time it forms that, it causes this RNA to get destroyed. Okay. All right, we'll finish off this on Wednesday for sure. I put the little song on D2L if you want to listen to it. So.